good morning all. Okay, in the last week we have started one lesson, biology and human welfare. We completed 3 days up to now. In the first class, we learned contributions of Dr. Ella Prad Subbarao to biochemistry and medicine. Okay. Okay. After that, we learned what is parasitism, what is meant by parasitism, okay, what is parasite, what is host. The next types of parasites based on the interaction between the host and parasite, ectoparasite, endoparasite, hyperparasite, monogenetic parasite, digenetic parasites. We learn those types of parasites. Okay, after that, we learn types of host, primary host, secondary host, reservoir host. Primary host is also called definitive host. Secondary host is also called intermediate host. What is the primary host for Plasmodium and the secondary host for Plasmodium? Primary host for Eukarya, secondary host for Eukarya, and primary host for Fasciola hepatica, secondary host for Fasciola hepatica, and reservoir host for Plasmodium, reservoir host for Trypanosoma gambiense. Okay, we learned those all things in the previous classes. After that, we learned types of vectors, biological vector, mechanical vector, and examples for biological vector and mechanical vector. After that, we learned parasitic adaptations, anchoring, and obligate envelopes, and facultative envelopes. Okay, development of production of anti-enzyme. Okay, having the protective cuticle and tegument, and reproductive potential, complexity of the life cycle, especially in the endoparasites, smart parasites, polyembryony. Okay, we discussed those all things in parasitic adaptations. After that, we learned effects of parasites on host. Okay, parasites make hosts unavailable. They make them weak. They make their bodies weak, and they make the nutrients unavailable for the host and metabolites also. And we learned neoplasia, hyperplasia, hypertrophy, parasitic castration, very important. 2 marks one. Good morning. KS Mallikarjuna, good morning. Shankar Noy, good morning. Gurdesh, good morning. Okay, next hyperplasia, hypertrophy, gigantism in case of snake. And next Leishmania donovani, Leishmania tropica. Okay, Leishmania tropica, Leishmania donovani. Okay, Leishmania donovani, Leishmania tropica, Trypanosoma gambiense. Okay, it causes sleeping sickness. Okay, Eukarya. Okay, we discussed these all things in the previous class. Okay, come to today's topic is Entamoeba histolytica, life cycle of Entamoeba histolytica. As I told you in the previous part, from this lesson, okay, our weightage from this lesson is 14 marks in final exam. One essay will be given. This is the first essay. Okay, we have Ascaris life cycle, Plasmodium, man and mosquito, 2 essays. Total we have in this lesson for nearly 5 important essays: Eukarya, Ascaris, Entamoeba, Plasmodium life cycle in man and mosquito. Okay, these are the very important essays. Some more are the most important are these 5. Okay, every year one question is given from these 5 essays. Okay, this is the first essay from this lesson. Life cycle of Entamoeba histolytica. Okay, life cycle of Entamoeba histolytica. Let us start. Okay, this is our today's topic, life cycle of Pentamoeba histolytica. If some more students set to join. Okay, link was sent to your group. Okay, link was sent to your group. Open your group, boys and girls groups. You will find the link. Open that link. Okay, right. Okay, Entamoeba histolytica. What is classification? First, let us study the classification of Entamoeba histolytica. Okay, this is the systematic position of or taxonomic position of Entamoeba histolytica. It belongs to the phylum Protozoa. Entamoeba histolytica belongs to the phylum Protozoa, and its subphylum is Sarcomastygopora. Subphylum Sarcomastygopora, and class is Rhizopodia. Okay, this is the classification. Okay, this is the phylum Protozoa, subphylum Sarcomastygopora, and class Rhizopodia. Can you mention what is our class? Can you type in our class? Which class do we belong? Which class do we belong? Which class do we belong? Okay, right, very good. Which subphylum do we belong? Raju, good morning, and Venkateswara, good morning. Correct answer. Okay, primates. Raju, it is primata is the order, not the class. Okay, now my question is, which subphylum do we belong? Which subphylum do we belong? Okay, Shankar, right answer. Which subphylum do we belong? Which subphylum do we belong? Okay, right, very good. Vertebratum. Which phylum we belong? You know, that is Chordata. Okay, now we are comparing our systematic position and the systematic position of Trypanosoma Entamoeba histolytica. Okay, its phylum is Protozoa. Our phylum is Chordata. Subphylum is Sarcomastygopora. Our subphylum is Vertebrata or Craniata. Class is Rhizopodia. Our class is Mammalia. Okay, Entamoeba histolytica. Okay, is microscopic. That means it is very small. We study protozoans in microbiology. Okay, these organisms are they are not visible to our naked eye. We have to see using microscope. And it is a monogenetic parasite. Monogenetic parasite. You know the meaning of the monogenetic parasites. Okay, those who do not know, I repeat again. The parasite which completes its life cycle in the single host. It is called Ascaris is a monogenetic parasite. Entamoeba is a monogenetic parasite. Pinworm, hookworm. Okay, these are the monogenetic parasites which complete their life cycle in single host. Next one, disease. What is the disease caused by Entamoeba histolytica? Okay, Entamoeba histolytica causes a disease called amoebiasis. Okay, amoebiasis, or we can call amoebic dysentery. Amoebic dysentery or amoebiasis is the disease caused by the Entamoeba histolytica. Where does it live? Where does it live? It lives in mucosa and submucosa of the large intestine. Mucosa and submucosa. What is meant by mucosa and submucosa of the large intestine? Okay, you will learn in the intermediate second year. In 10th class, you learned about our small intestine, large intestine. If you take the transverse section, I am briefly explaining here what is mucosa, submucosa. This is the transverse section of large intestine. Okay, you know large intestine. You learned about large intestine in 10th class also. Okay, this is the colon, ascending colon. This is transverse colon. This is the descending colon. Then rectum, and finally there is an anus. Okay, if I take a section here, I am cutting a section here, transverse section. TS of leaf you learned in 10th class. Transverse section of leaf. I am taking the transverse section of large intestine. And this is the outermost layer. Okay, this is called serosa. After this one, there is a layer of, there are muscles. Okay, there are muscles, longitudinal smooth muscles. A layer of longitudinal smooth muscles. After this one, there are circular muscles. First one, longitudinal muscles here. Okay, these are smooth muscles. Smooth muscles are arranged in two layers. One layer is arranged lengthwise like this. One layer is arranged like this one in a circle manner. Okay, these are the circular muscles. These are the longitudinal muscles or smooth muscles. After this one layer comes after this one. Okay, this layer is called submucosa. This is called submucosa. Okay, innermost layer is called mucosa. This layer is called, this is submucosa, and this layer is mucosa, and this is submucosa. And what is this space? This is called lumen. That means this space. Okay, this is the lumen. Buffering. Okay, no buffering here. Everything is fine here. Syed. Okay, what is this one? Submucosa, and what is this one? Mucosa. Okay, it leaves entamoeba histolytica. For example, if a person has entamoeba histolytica in his body, especially trophozoite state, where does it live? Okay, the body, for example, where is our heart? Okay, we can say, where is our kidney? You know the location. Where is our brain? You know the location. But this entamoeba histolytica during the especially trophozoite state, where does it live in our body? The location where it lives is mucosa and submucosa of large intestine. This is the place where it inhabits or where it lives. Okay, it lives in mucosa and submucosa of the large intestine. Next one, what is meant by plasma lemma? Okay, if you observe this diagram, this is the trophozoite stage. The body of entamoeba histolytica is surrounded by, this is called plasma lemma. This is called plasma lemma. The body of entamoeba histolytica is surrounded by, is it okay now? Okay, the body of entamoeba histolytica is surrounded by a layer. That layer we call it plasma lemma. Like our body is surrounded by skin. Like that, its body is covered by layer that is called plasma lemma. Next, if you go inside this plasma lemma, there is a cytoplasm. Okay, there is a cytoplasm inside. Okay, this cytoplasm is differentiated. The cytoplasm is differentiated. The cytoplasm, okay, right? The cytoplasm is differentiated into, okay, this is called outer ectoplasm. Okay, you can observe here, I am drawing again here, labeling here. This is called ectoplasm. The cytoplasm which is outside, this is called just below the plasma lemma, this is called this white part. This is called ectoplasm. Okay, ectoplasm. How is the ectoplasm? Ectoplasm is outside, ectoplasm is thin, ectoplasm is clear, and no granules are here. It is a non-granular layer. Non-granular. Okay, that's about ectoplasm. Ectoplasm is outer, ectoplasm is thin, ectoplasm is clear, ectoplasm does not have granules, it is non-granular. For example, you know WBC, blood cells. White blood cells are two types, granulocytes and agranulocytes. Granulocytes have granules in the cytoplasm. Agranulocytes do not have granules. Granules means small, grain-like structures in the cytoplasm. Okay, here those are not present here. Non-granular. Endoplasm. Endoplasm here, if you observe endoplasm, this is the endoplasm, and this dotted portion, endoplasm, it is inner. This is endoplasm is inner. Okay, this portion is the endoplasm. The endoplasm is, how is the endoplasm? Inner, inside this one, and it is thick and it is granular, opposite to this one. Okay, those are the difference between ectoplasm and endoplasm. Okay, next let us talk about the pseudopodium. Pseudopodium of the Entamoeba histolytica. If you take amoeba, amoeba has many pseudopodium, polypodium. But here only one pseudopodium is present. Okay, one special name is given to this one. Okay, pseudopodium is called monopodial. This is the pseudopodium, this part. Okay, this is the pseudopodium. It is called monopodial because only one pseudopodium. If you take amoeba, amoeba has, this is one pseudopodium, this is one pseudopodium, this is one pseudopodium, many are there. But here only one pseudopodium, that too in the anterior side. Okay, that's why it is called monopodial. Only one pseudopodium. One pseudopodium is present, that is present, develops on the anterior side. Next one, it moves in a straight line. Important. It moves in a straight line because it has only one pseudopodium. And this pseudopodium is named as logopodium because it has a blunt end. If you take our finger does not have. Sir, doesn't it move like amoeba? No, it cannot move like amoeba. Amoeba has, it is polypodial. Many pseudopodia are present, but it has only one pseudopodium. Okay, for example, if you have one leg and if we have two legs, is there any change in our way of locomotion? Definitely there is a change. Okay, when two legs are, we move in one way because it has only one pseudopodium. It moves in one direction. Okay, it is called, that pseudopodium is called logopodium because it has a blunt end, not pointed. It has a pointed end, but the finger has a blunt end. Okay, next, organelles. Inside this endoplasm, there are different organelles are present. But is it eukaryote or prokaryote? Can you type in your answer? Buddhish and remaining Shankar, Venkateswarlu and Sayed. Okay, yes, you have to draw the diagram. Okay, later evening time or later you can draw the diagram. Okay, this is the 4 marks question answer. Write about trophozoite stage. Write about trophozoite stage. That is the 4 marks question. Okay, here this is the endoplasm. Is it eukaryote or prokaryote? Type your answer. Is it eukaryote or prokaryote? Is it eukaryote or prokaryote? Is it eukaryote or prokaryote? It is unicellular eukaryote. Okay, very good. It is unicellular eukaryote because it has a definite nucleus is present. Now we are talking about tropozoites, right? Eukaryote. Okay, again I repeat, Entamoeba histolytica. Entamoeba histolytica is a microscopic. It is monogenetic because it completes the life cycle in a single host. How it causes a disease called amoebiasis or amoebic dysentery. It is transmitted from one person to another person. Okay, mode of infection is through contaminated food and water. Through contaminated food and water, that contaminated food may have an infective stage. You will let that one later tetranucleate stage and tarvata vastuni. Next, mechanical vectors. The mechanical vectors are housefly and cockroaches. Mechanical vectors are housefly and cockroaches. Earlier we discussed what is mechanical vector and biological vector. Earlier we discussed. Okay, it is a mechanical, it is spread by mechanical vector. Mechanical vectors, we learned two examples. One is a housefly, another one is called cockroach. What is meant by mechanical vector? The vector which transmits, which transfers the disease causing organism from one host to another host. No development takes place. Okay, no development of the pathogen takes place in the vector. That vector is called mechanical vector. For Entamoeba histolytica, the mechanical vectors are mechanical vectors. One is housefly. I am erasing this one. Phylum, subphylum, and class. The mechanical vectors are housefly. Housefly, scientific name Musca domestica. Housefly, scientific name Musca domestica. Musca domestica. Housefly, scientific name Musca domestica. Musca is the genus, domestica is the species name. It belongs to the phylum Arthropoda. Another one is cockroach. Cockroach scientific name Periplaneta americana. Okay, later we study the cockroach, one of the chapter. The cockroach scientific name Periplaneta. Just you write cockroach scientific name, you can learn it later. Okay, here, it is more microscopic monogenetic parasite causes a disease called amoebic dysentery. The mechanical vectors of cockroach are Entamoeba are cockroach and housefly. The scientific name of housefly is Musca domestica and cockroach Periplaneta americana. In the life history of Entamoeba histolytica, there are three stages. First, I am talking about one stage. This stage is called trophozoite stage. Trophozoite stage. Okay, you know Corona, COVID virus. COVID virus is present all over the world. It is a pandemic disease. It is not confined to just one continent or one country. It is present all over. It is prevalent. It is common. It is present everywhere. It is a pandemic. But this disease, amoebiasis or amoebic dysentery, this disease is more common in tropical regions and subtropical regions. Tropical regions means you might, in social studies, you might learn, between the Tropic of Cancer and Capricorn. Okay, that is the tropical region on either side of the equator between the Tropic of Cancer and Capricorn. And subtropical regions, above the Tropic of Cancer and below this Tropic of Cancer. In those areas, it is very common, and especially in the densely populated areas where the population is very. For example, recently we learnt about Dharavi in Mumbai. People are very crowded, densely populated areas. In those areas, it is very common, especially in the rural areas. Also, this disease is very common in those areas. Okay, now we are going to talk about trophozoite stage. In the life cycle of Entamoeba histolytica, it passes through three stages. Okay, in our life, there is a childhood stage, there is an adolescent stage, there is a middle age, old age. Like that, we pass through different stages. Okay, here also, there are mainly three stages are there. One is trophozoite. This is a diagram which represents trophozoite stage. What are the remaining two stages? Pre-cystic stage and the cystic stage. Those are the remaining two. Here I am writing the three stages. One is trophozoite stage. Next one is pre-cystic stage, and the third one is cystic stage. These are the three stages in the life history of entamoeba histolytica, right? Okay, the pre-cystic stage and cystic stage. Okay, now we are going to talk about trophozoite stage. Okay, this evening you have to learn. I will send you this 4 marks one. Okay, I will tell you. I will send the question and answers of this lesson. You simultaneously prepare objective as well as IP questions. Okay, if you prepare simultaneously, okay, you can get both MCQ as well as IP questions. Okay, this is the 4 marks question in this lesson. Okay, describe the structure of trophozoite. On the entamoeba life cycle, we have 4 marks questions. We have nearly 6 2 marks questions and one essay. Okay, you have to, if you prepare this topic and we histolytica, you are able to write essay, you are able to write 4 marks, you are able to write to all 2 marks also. Okay, trophozoite stage, pre-cystic stage, and cystic stage. Now we are talking about trophozoite stage. In trophozoite stage, I told you where does it live? It lives in the mucosa and submucosa. It is the motile stage. Trophozoite stage is motile stage. What is meant by motile? Remaining two stages. This is a non-motile. It is a non-motile. It is also non-motile, and it is motile. Not only motile, it is more active. Motile means have the ability. Send the notes of today's class. Okay, definitely. Okay, this is motile. That means able to move. For example, sperm. Sperm has head. Sperm has neck. Sperm has middle piece. Sperm has tail. It can swim. It is motile. On the other hand, the worm is stationary, non-motile. Like that, like worm pre-cystic state, we have to write notes what you sent in group. Yes, you have to note down your own handwriting. You have to write down, and a test also, sir. Okay, this evening, okay, on yourself write that 4 marks question and send me answers. You write down in on paper and send those answers to me. I will check it. I will type my number here. Okay, this evening your work is okay, learn the structure of trophozoite. I will tell you some more 2 marks at the end of this topic. Okay, read 4 marks and 2 marks also I will tell you at the end of this lesson. Okay, you have to send me, you have to write that 4 marks and send to that number. Okay, trophozoite stage, pre-cystic stage and cystic stage. This is the motile stage. It is motile stage. These two are the non-motile stages. And it is motile stage because it has pseudopodium. It has pseudopodium called logopodium. But in these two stages, pseudopodium is absent. That means legs are absent. When legs are absent, they cannot move. Okay, they can move, but motility is less. But here, okay, these two do not have pseudopodium. That's why they cannot move. and then it has pseudopodium. That's why it can move. It is a motile state. Next one, where do they live? Okay, these are two stages. They live in the lumen. They live in the lumen. This place, they live in lumen in this place. But this state, trophozoite state, it lives in mucosa and submucosa. This is the living place of these three stages. I repeat again. ట్రోపోజాయిట్ స్టేజ్ మన బాడీలో ఎక్కడ ఉంటుంది, ఎవరికైతే ఈ డిసీజ్ ఉంటుందో వాళ్ళ బాడీలో అది ఎక్కడ ఉంటుందంటే మన ఇంటెస్టైన్ వాల్ లో మ్యూకోసా అండ్ సబ్ మ్యూకోసాలో ఉంటుంది. రిమైనింగ్ టూ స్టేజెస్ ఎక్కడ ఉంటాయంటే దిస్ ఇస్ మన ఎలిమెంటరీ కెనాల్ మధ్యలో మన ఫుడ్ వెళ్ళే రంధ్రం ఉంటుంది, ఫుడ్ పాస్ దట్ ఈస్ కాల్డ్ లూమెన్ అంటాం. ఆ లూమెన్ లో ఉంటాయి దీస్ టూ లివింగ్ lumen and lumen. This one is present in submucosa and mucosa. Next one, trophozoite stage is called pathogenic stage. Pathogenic stage. What do you mean by pathology? I told you earlier, study of disease. It is a pathogenic stage, means it is able to cause disease. It is able to cause ulcers in the intestine, okay, by destroying our tissues. It is a pathogenic stage. It is a dangerous stage. It is a very harmful stage in our body. Okay, it is a pathogenic stage. That means it has the ability to cause the disease. Next one, the body of trophozoite is surrounded by plasma lemma. Inside that one there is a cytoplasm. The cytoplasm is differentiated into outer ectoplasm and inner endoplasm. Differences between ectoplasm and endoplasm: it is outer, it is inner, it is thin, it is thick, it is clear and non-granular, but it is granular. It has so many granules like structures are present here. Here it is granular, it is non-granular. These are the difference between ectoplasm and endoplasm. Now come to pseudopodium. It has only pseudopodium. నేను చెప్పాను ఇందా మీకు, ఈ రెండింటిలో pseudopodium ఉండదు అసలు. there is no locomotor structure. That's why they are non-motile stages. Okay, pseudopodium, it is monopodium because all three are pseudopodium is present, unlike amoeba. And one pseudopodium, it moves in a straight line. And next one, lobopodium. This pseudopodium is called lobopodium. It is named, it is given the name lobopodium because it is like a finger-like blunt. Okay, next one, organelles. What are the organelles present? Okay, if you take our body cells, okay, they have ribosomes, endoplasmic reticulum, RER, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus. Okay, like that, many organelles. If you take plant cells, there is a chloroplast, there are lysosome. Okay, lysosome, suicidal bags of the cell. These all are cell organelles. What type of organelles are present in the trophozoite state? Which organelles are absent? Okay, let us see that. Organelles, nucleus is present. Very, very important one, nucleus. Here, the nucleus is cartwheel shaped nucleus. Ante bandi chakram lanti nucleus. That is one of the important characteristic feature of trophozoite state. Presence of cartwheel shaped nucleus. అంటే బండి చక్రం లాంటి న్యూక్లియస్ ఉంటుంది. ఇవన్నీ స్పోక్స్ లాగా, క్రోమాటిన్ అనేది స్పోక్స్ లాగా అరేంజ్ అయి ఉంటుంది. Presence of cartwheel shaped nucleus is present. What type of nucleus? A nucleus is vesicular and cyclic, vesicular and cartwheel shaped nucleus. Very, very important point. Cartwheel, cartwheel shaped nucleus is present. అంటే బండి చక్రం లాంటి nucleus ఉంది. ఇవన్నీ spokes లాగా, ఇది chromatin. And this is inside the nucleus there is a nucleolus. Inside the nucleus there is a nucleolus is present. That's about nucleus. Come to the ribosomes are also present. Here I shown you this red black color one. Okay, these are ribosomes. Ribosomes are involved in. Can you type in your answer? Ribosomes are involved in. Quickly type in your answer. Ribosomes are involved in. Ribosomes are involved in. Ribosomes are involved in Lakshmi Devi. Okay, right. Very good. Protein synthesis. Ribosomes are involved in protein synthesis. Okay, here ribosomes and food vacuole also present. Food vacuole here very important part. Here two types of food vacuoles are present. I am labeling here. Okay, this is the one food vacuole with bacteria. Food vacuole with bacteria. Okay, where is that bacteria? For example, in our body there are a lot of bacteria in our gut. There are symbiotic bacteria, useful bacteria, not harmful bacteria. Our intestine has one flora that is called a bacteria. Huge, there are lakhs of bacteria are present in our intestine. That means that our intestine also, it is a habitat for some bacteria. I told you earlier, habitat, the place where an organism resides or lives is called habitat. Our intestine is also a habitat for many useful bacteria like E. coli, Escherichia coli. Those bacteria are useful in, they help in producing bacteria, some of the vitamins. They produce vitamin. అదేం చేస్తుందంటే ఈ ట్రోఫోసైట్ స్టే మన లోపల మన ఇంటెస్టైన్ లార్జ్ ఇంటెస్టైన్ లో ఉన్న యూస్ఫుల్ బ్యాక్టీరియాని కూడా తినేస్తుంది. దాన్ని ఆ బ్యాక్టీరియాని ఏమంటారు? వి కాల్ ఇంటెస్టైనల్ ఫ్లోరా అంటాం. ఫ్లోరా అండ్ ఫానా. ఫ్లోరా మీన్స్ ప్లాంట్స్, ఫానా మీన్స్ యానిమల్స్. Intestinal flora is there. That intestinal flora, that means bacteria, and those bacteria are also ingested by this trophozoite. That's why this is the food vacuole. This is the food vacuole. It is a food vacuole. Food vacuole with bacteria. Food vacuole with ఈ బ్యాక్టీరియా మన బాడీలో ఎక్కడ వచ్చాయంటే, మన బాడీలో దేర్ ఆర్ సో మెనీ యూస్ఫుల్ బ్యాక్టీరియా ఉంటాయి ఇంటెస్టైన్ లో. ఒక ఆ యూస్ఫుల్ బ్యాక్టీరియాని కూడా తినేస్తుంది. నెక్స్ట్ వన్, దేర్ ఇస్ అనదర్ టైప్ ఆఫ్ food vacuole is present here. Okay, that food vacuole with the RBC, red blood cell. Okay, this is the food vacuole. This is the food vacuole. This is food vacuole with the red blood cell. This is another characteristic feature. This is another మనకి ట్రోఫోని ఐడెంటిఫై చేయడానికి ట్రోఫోసైట్ ని. Okay sir, it is every cell is, in every cell it is present. Okay, whether you are asking, Karthik, good morning. Okay, these are the vacuoles. Okay, these are the vacuoles. These are the food vacuole. This is the vacuole. Blue color line to draw చేసిన food vacuole. దాని లోపల ఇది RBC. అంటే మన RBC ని తినేస్తుంది. Okay, these are the RBC. Food vacuole. These are the food vacuoles with RBC. Karthik, you are from MPC. I think. What's your doubt? I did not get it. Okay, right. The organelles completed. Which organelles are present here? There is a convolution-shaped vesicular nucleus is present. Second one, there are ribosomes are present, which are involved in protein synthesis, and there are food vacuoles are present. Some of the food vacuoles are with bacteria, and some of the food vacuoles are with red blood cells, RBC. RBC with nucleus. Yes. Some of the vacuoles have. Is it present in every cell? Okay, in every cell. Okay, in every cell means trophozoite stages. అన్నిటి కూడా, ఎందుకంటే tropho means food. Autotroph, heterotroph. Troph means food. Auto cell. This is the trophozoite stage. It is a feeding stage. తినేస్తుంది. ఎవరు తింటుందంటే intestine లో submucosa ఉన్న cells, epithelial cells. It feeds on our epithelial cells. It feeds on our bacteria. It feeds on our RBC. వీటన్నిటిని తినేస్తుంది. కాబట్టి కొన్ని సమ్ ఆఫ్ ద అన్ని ట్రోఫోజైట్ ఎందుకంటే దానికి ఫుడ్ అది. ఓకే, మోస్ట్ ఆఫ్ ద ట్రోఫోజైట్ స్టేజెస్ have these RBC in them. Okay, next one is organelles absent. Which organelles are absent in the trophozoite state? Which organelles are absent? Some of the organelles we cannot observe here, not present here. What are the contractile vacuole is absent? Contractile vacuole is absent here. Contractile vacuole is absent. Contractile vacuole you learned in 10th class. What is the significance of contractile vacuole? Karthik, can you tell me what is the significance of contractile vacuole? Lakshmi, what is the significance of contractile vacuole? What would happen if contractile vacuole is not present? You learned in 10th standard. Quickly. Contractile vacuole is a osmoregulatory organelle. Okay, it helps in osmoregulation, maintaining water and salt balance in the body. But it is also a protozoan like amoeba. Okay, entamoeba, that is amoeba, both are protozoans, but this one does not have contractile vacuoles, but yes, they maintain ionic balance. Yes, Mallikarjun. Okay, but on the other hand, the other one, okay, amoeba has contractile vacuole. Contractile vacuole maintain osmoregulation. It lives in fresh water, but it is living in our intestine. It is an enteric parasite. Enteric parasite means live in the gut. Okay, this enteric parasite, as it living in our body, okay, the conditions are different out in the water and here. Okay, that's why here contractile vacuole is absent as it is living in a isotonic medium. I will tell you details about what is hypertonic medium, hypotonic medium, isotonic medium. For example, one animal is living in water. Okay, here is water. In this water there is one animal. The concentration of salt here and the concentration of salt in the cytoplasm and concentration of the salt in the water is same. That means this animal is living in a isotonic medium. No problems here, because if the salt concentration is same on both sides, it does not have any problem. Okay, contractile vacuole is important to maintain, to remove the excess water from the body లో నుంచి వాటర్ తీసేయడానికి హెల్ప్ చేస్తుంది. ఓకే, బికాజ్ అమీబాలో ఎందుకు ఉందంటే అమీబాలో సైటోప్లాజం ఇస్ హైపర్టోనిక్. It is only lives in small intestine, sir. No, it lives in large intestine, not small intestine. Cascaris lives in small intestine, Lakshmi Devi, but this one it lives in large intestine, not small intestine. Large intestine lo mucosa and submucosa layers lo untundi. Okay, large intestine lo ఉన్న solution అక్కడ ఉన్న medium కి, okay, దీనికి గంటమీప histolytic cytoplasm కి ఉన్న ఇద్దరు osmolarity సేమ్ గా ఉంటుంది. కాబట్టి దీని contractile vacuole is not necessary. That is important point. It is not mentioned in our test book. Okay, only in large intestine. Okay, later, okay, in the initial stages in the large intestine, later secondary stages it go to other body parts also. In the initial stages, in the primary stage, okay, it only confined to large intestine. Later it may go to through the blood circulation, it may go to other body parts as well. Okay, contractile vacuole is absent as it is living in isotonic medium. This is not given in our textbook, which is very important point. It is as it is living in isotonic medium. Okay, contractile vacuole is not necessary to this one. Trophozoite stage lo contractile food vacuoles are present. Food vacuoles are, but contractile vacuoles are absent. Next, mitochondria are absent. This is one 2 marks question. Okay, why mitochondria are absent? That's why it is an obligate anaerobe. I told you in parasitic adaptations, there are some facultative anaerobes as well as there are some obligatory anaerobes. Okay, it must perform anaerobic respiration. I told you in day before yesterday's class. Okay, cellular respiration is of two types. Cellular respiration is of two types: aerobic respiration and anaerobic respiration. For aerobic respiration, mitochondria is required, but here mitochondria is absent. As mitochondria is absent here, it is an obligate anaerobe. Very, very important. It is a 2 marks question. It is an obligate anaerobe. Obligate anaerobe because mitochondria is only one line answer here. Why entamoeba histolytic is an obligate anaerobe? Due to the absence of mitochondria, it must perform anaerobic respiration. Okay, right. Next one, histolysine. Why the name? Okay, entamoeba histolytica ki histolytica ane peru vachindante. What is meant by histology? 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 Okay, what is meant by histology is the study of tissues. Here, this one, okay, entamoeba histolytica. Entamoeba histolytica, it is a dissolving our tissues. It is dissolving our tissues present in mucosa and submucosa. How can it dissolve our tissues? Because it is capable of producing. Histolysine. Okay, that's why the name entamoeba histolytica ki histolytica ane species name enduku ichcharante because it can produce histolysine enzyme. Okay, this enzyme, the histolysine, histolysine can damage our mucosa and submucosa, and it may cause ulcers, and bleeding also occurs in that particular part where those tissues are damaged. Okay, that is the reason why the name histolytica is given to, the species name histolytica is assigned to entamoeba histolytica because it is producing histolysine which can damage mucosa and submucosa tissues there. Next one, it is endoplasmic reticulum is also absent, and Golgi complex is also absent. What are the organelles? 4 organelles are absent here. We mentioned here contractile vacuole is absent because it is living in isotonic medium. Mitochondria is absent, that's why it is obligate anaerobe, and endoplasmic reticulum and the Golgi complex are also absent. Okay, next one, what are the characteristic features? దీన్ని మనం trophozoite ని ఎలా ఐడెంటిఫై చేయొచ్చు? How can we identify trophozoite in a person's stool? For example, there is a person who is suffering from amoebiasis disease, that means amoebic dysentery. In his stool, in his motion, in his stool, fecal matter, these stages are present. Pre-cystic stages are present. Cystic stages are present. If the person who is suffering from this one, there will be abdominal pain, irregular bowel movements. Okay, there will be mucus in the fecal matter, and there will be blood in fecal matter. వీటిని మనం రక్త విరోచనాలు అంటాం, బ్లడ్. మరి ఆ పేషెంట్ ని డాక్టర్స్ ఏం అడ్వైజ్ చేస్తారు? టు గో ఫర్ స్టూల్ టెస్ట్ అంటే మోషన్ టెస్ట్ చేస్తారు. ఆ ఫీకల్ మ్యాటర్ స్టూల్ ని టెస్ట్ చేసినప్పుడు అతనికి ఈ స్టేజెస్ కనపడితే, అంటే ఈ స్టేజెస్ అని ట్రోపోజైట్ స్టేజ్ ని మనం ఎలా ఐడెంటిఫై చేయాలి? 3 characteristic features are present. This is important one. I will show you here. What are the 3 characteristic features of trophozoite state? Okay, trophozoite stage have 3 characteristic features here. Characteristic feature one is cartwheel like nucleus. Cartwheel like nucleus అంటే బండి చక్రం లాంటి nucleus. Second one, chromatin like spokes of wheel. Cycle చక్రం లాగా spokes అంటే chromatin మెటీరియల్ ఇలాంటివి ఉంటాయి. Chromatin like spokes of wheel. Chromatin is like spokes of wheel, And okay, nucleus is like cartwheel. Okay, nucleus is like cartwheel. Chromatin like spikel spokes. Next one, food vacuole. Food vacuoles with the human RBC. Food vacuole with human RBC. Food vacuole with bacteria. Food vacuole with bacteria. And food vacuole with bacteria. And also epithelial cells. Epithelial. Okay, by these characters, we can identify it is a trophozoite state. Okay, then one trophozoite stage and మనం identify by those features. Okay, this is about trophozoite state. The next stage is pre-cystic state. Then after that there is cystic state. Okay, now I would like to ask you 2-3 questions from this completed topic. Okay, now why it is called, why it is obligate in row, why entamoeba histolytica is obligate in row, that is 2 marks. Just you one word you have to write, why it is obligate in row, why it is obligate in row, due to the absence of, why entamoeba histolytica is obligate in a row, due to the absence of. Okay, very good, you got it, 2 marks. Mitochondria, due to the absence of mitochondria. Why contractile vacuole is absent, as it live in which medium? Okay, very good, Raju, Udesh and Syed Karimullah. Why contractile vacuole is absent? Okay, right, Mallikarjun, Kusma. First time I am seeing Kusma here. Okay, Karthik. Okay, right, very good, Karthik, give right answer. Contractile vacuole is absent because it is living in isotonic medium. Very good, isotonic medium. Mallikarjun, okay, right, very good. Raju, Lakshmi Devi, it is living in isotonic medium. What are the characteristic features by which we can identify trophozoite? What are the, I am Vamsi, okay, right, Vamsi, okay, right, sorry. What are the characteristic features? What are the characteristic features by which we can identify trophozoite stage? What are the characteristic features by which we can identify trophozoite stage? One is cartwheel shaped nucleus. The other one is chromatin is like a cycle spokes. Chromatin is like, okay, right, very good. Okay, next one. Third one is, okay, vacuoles, food vacuoles with red blood cells. Okay, these are the 3 important characteristic features by which we can identify trophozoite stage from the pre-cystic and cystic stage. Okay, now I am moving on to the food vacuole with RBC, right? Food vacuole with RBC and cartwheel shaped nucleus. Okay, now let us talk about pre-cystic stage. But before the formation of cyst wall, that is called pre-cystic stage. Vacuole with RBC and bacteria, right? Vacuole with RBC, epithelial cells, and the next one, pre-cystic stage. Okay, this stage is like a spherical, like a ball, or in the oval shape. It is in oval shape, but no pseudopodium in oval shape or in spherical shape. And here there is a nucleus is present. A nucleus is present. And here there are some chromatoid bars are present. Chromatide bars. This is the reserve food material which are made up of ribonucleoprotein. Chromatide bars. This is the reserve food material because it is a non-feeding state. It is not a trophozoite state. Non-feeding food em tinadu mana body lo kabatti than local food. What is the reserve food in the plants? What is the reserve food in the plants? Okay, thank you, Karthik. What is the reserve food in the plants? In the leaves of the plant, they store the excess food in the form of what is the reserve food? What is the reserve food? Plants convert the excess glucose into insoluble form called. You have done one experiment also to prove that to prove the presence of dash in the leaves by using iodine reagent. Now you got it, I think. Right, very good. Mallikarjun, starch. Okay, starch is the reserve food in the plants. When during the daytime they perform photosynthesis, when there is excess glucose, the glucose is converted to starch. Starch is insoluble, whereas glucose is soluble. They convert soluble glucose into insoluble starch and and store in the leaves and other parts of the plant body. Here, the reserve food here is glycogen granules. There are some glycogen granules. Glycogen is also called anyone. Glycogen half-life experiment, moles half-life experiment, that is not that. Moles half-life experiment is to prove that CO2 is necessary for photosynthesis, not starch. For starch experiment, to prove the starch, you keep the so you keep the leaf in the boiling solution, then it becomes pale color. Then you pour iodine solution. Then if there is starch, it turns into deep blue color. Okay, moles half-life experiment, that is to prove that CO2 is necessary for photosynthesis by taking KOH solution in that one. The body. And what do we call? Okay, glycogen is also quickly. I want quick response. Our time is running out. Glycogen is also called. Very good, Karthik. Okay, there you are. Animal starch. Glycogen is called animal starch. Okay, in these animals there is a glycogen granules are present, chromatide bars are present. This is the cytoplasm here. Okay, this is the diagram of pre-cystic state. Here, these are the glycogen granules. Which is the reserve food. Chromatide bars. Bar means like a, buy 5-star chocolate like a bar, soap bar. Like that is chromatide bars made up of ribonucleoprotein. Made of, very important one, ribonucleoprotein, which is the reserve food. Okay, these two are the reserve food because it is a non-feeding stage, non-pathogenic stage, non-motile stage. It cannot move because the lobopodium is absent. It does not have food vacuoles. No food vacuole here. It cannot take the food. Okay, that's why it has to store the food. Okay, that reserve food is in the form of glycogen granules and chromatide ball. Okay, these are present. Okay, repeat again. Pre-cystic stage. Okay, pre-cystic stage. Okay, it is a non-motile. It is a non-motile stage. Non-pathogenic stage. and which is like oval or spherical shape, that is not important. Reserve food. The reserve food is in the form of, one is glycogen granules. The other one is chromatide bars. Chromatide bars are made up of ribonucleoprotein. Next one, it lives in the previous in the chapter, it is non-motile, already completed. It lives in the lumen of large intestine. And now no food vacuoles are not present. It is a non-pathogenic stage. Okay, these are the points related to. Okay, these are the point. Motile means mobile, able to move. Okay, pre-cystic state. It is a non-motile because no logopodium, pseudopodium, and logopodium is not present. Next one, it is non-pathogenic. It does not cause any harm. It cannot destroy our tissues because it is living in the lumen, not in the wall. And it has a reserve food. It is a non-feeding stage. I have written that point here. Okay, it is a non-feeding stage. Non-feeding stage. Feeding stage means the tintundi ante trophozoite stage, feeding stage. Okay, reserve food, glycogen granules, chromatide bars, and it lives in the lumen of the large intestine. I showed you earlier, and it is a non-pathogenic stage and non-feeding stage. That's about the pre-cystic stage. Now come to the last stage, that is cystic stage. In tomorrow's class, I will complete cystic stage. And also remaining part, this life cycle of Entamoeba histolytica will be completed in tomorrow's class. Wait. Okay, I have sent you some questions from this IP material. Okay, we have here, describe the structure of trophozoite of Entamoeba histolytica. Okay, this is the 4 marks question from this lesson, and 2 marks I have sent you. Okay, before my class I have sent you question and answers from this lesson. Okay, what we have learned in today's మనం నేర్చుకున్న క్లాస్ లో కొన్ని క్వశ్చన్స్ ఉన్నాయి ఇక్కడ. What are the 2 diagnostic features of trophozoite of Entamoeba histolytica? What are the 2 diagnostic features, identifiable features of trophozoite of Entamoeba histolytica? Chromosome and chromatids. Difference between chromosome and chromatide. What is chromatide? Chromatin. Okay, you will come across the term in the botany during cell division. At some stage, at some point of time, we call it chromatin, and at some point of time, some point of cell division, we call it chromosome. Okay, chromosome, zome means bodies. Okay, the color, chrome means color. Google chrome. Chrome means color. Okay, you learned in 10th class urochrome, which gives color to our urine. Urochrome, the color of the urine is because of urochrome. Chromosome, chrome means color. Soma means body, color observing bodies. When they observe colors and they attain. Okay, right. What are the 2 diagnostic features of trophozoite of Entamoeba histolytica? Presence of RBC in the food vacuoles And cartwheel shaped nucleus are the characteristic features of trophozoites of Entamoeba histolytica. You got it. Entamoeba histolytica is an obligate anaerobe justified. Entamoeba histolytica, you know the answer, just one word. Okay, Entamoeba histolytica always exhibits anaerobic respiration due to the availability of oxygen is very rare to it, hence obligatory absence of mitochondria is the answer. Next one, what is the reserve food in the pre-cystic stage of Entamoeba histolytica? Type in your answer. What is the reserve food in the pre-cystic stage of Entamoeba histolytica? Okay, right, Karthik. What is the reserve food in the pre-cystic stage? It is my on the back. What is the reserve food material? Okay, this is IP material now. IP intermediate public exams material. Okay, right. Glycogen granules and chromatide bars. Okay, these are some of the questions, and there is another one: the difference between pre-cystic stage and cystic stage. Yes, glycogen granules. Okay, these questions I have already sent you. Okay, write down this evening. Practice those question answers. One, you have to send me. Okay, 4 marks question. Structure of trophozoite. You have to prepare and draw a diagram and send it to me. Okay, I have provided you with my mobile number there. What is the page number, sir? Okay, do you have material? If you have material, okay, నేను మీకు మార్నింగ్ ఈ క్వశ్చన్స్ పెట్టాను 3 pages లో. ఓకే, 4 మార్క్స్ క్వశ్చన్ మీకు పేజ్ నెంబర్ 62 లో ఉంటుంది మెటీరియల్. మీ దగ్గర ఉందో లేదో పేజ్ నెంబర్ 62. Describe the structure of trophozoite of Entamoeba histolytica. Next one, 2 marks questions are in the page number 50. Page number 56 లో ఉన్నాయి 2 marks questions. ఇక్కడ ఇవి మెటీరియల్ లో ఏ రోజు వర్క్ ఆ రోజు కంప్లీట్ చేస్తే మీకు ఈజీగా ఉంటుంది, నేర్చుకోవడానికి ట్రై చేయండి. Okay, Syed, if you have material, prepare it. Okay, goodbye to all of you. Thank you.